Hello friends, we are going to study over here about microbodies and their types. Microbodies are found in all eukaryotic cells. They contain oxidative enzymes such as catalase and urate oxidase at such high concentrations that in some cells the microbodies are seen in electron micrographs because of the presence of granular or crystalloid core that contains enzymes. Microbodies are found in cells of protozoa, fungi, plants, liver and kidney of vertebrates. They are spherical bodies of oxygen 1.5 micrometer in diameter and are found in close association of endoplasmic reticulum, mitochondria and chloroplast. Microbodies differ from mitochondria and chloroplast in many ways. They possess a single membrane and lack DNA as well as ribosomes. Microbodies are thought to acquire their proteins by selective import from the cytosol. As they have no genome, all of their proteins must be imported. Microsome resembles endoplasmic reticulum as they are self-replicating, membrane-enclosed organelle that exist without a genome of its own. Two main types of microbodies are found in nature. They are glyoxysomes and peroxisomes. Let us see both of them in detail. First, glyoxysomes. Glyoxysomes are specialized peroxisomes found in plants, particularly in the fat storage tissues of germinating seeds and also in filamentous fungi. As in all peroxisomes, in glyoxysomes the fatty acids are hydrolyzed to acetyl-CoA by peroxisomal beta oxidation enzymes. Glyoxysomes also participate in photorespiration and nitrogen metabolism in root nodules. The second one is peroxisomes. These are major sites of oxygen utilization. It utilizes molecular oxygen like mitochondria, but instead of having cytochromes and capacity of ATP synthesis like them, they contain flavin-linked oxides and catalase for hydrogen peroxide metabolism and also enzymes for fatty acid metabolism. An essential biosynthetic functions of animal peroxisome is to catalyze the first reactions in the formation of plasmalogens, which are the most abundant class of phospholipids in myelin. Let us have a look at history of peroxisomes. C.D. Dewey and P. Bauduin in the year 1966 termed peroxisomes for the microbodies of mammalian systems and also studied their structure and function in detail. Leaf peroxisome was first isolated from spinach leaf homogenate from mesophyll cells by Tolbert and et al. In the year 1968. Let us have a look at how isolation of peroxisome is carried out. Under electron microscope, catalase can be visualized when stained with DAB, that is 3,3-diaminobenzidine. It appears as a dark spot as it is electron opaque. For isolation, the tissue of liver, kidney, intestine, brain, etc. is homogenized carefully as it may break the single membrane bound peroxisomes. The separation is achieved by either sucrose density centrifugation or isopycnic ultracentrifugation. The palette obtained here contains peroxisomes. By density centrifuge, it can be identified due to a marker enzyme, catalase enzyme, as well as single membrane is another unique feature. Peroxisomes are unusually diverse organelles and may contain different sets of enzymes even in different cells of same organisms. They can also adapt remarkably to changing conditions. Yeast cells grown on sugar, for example, have small peroxisomes. But when some yeast are grown on methanol, they develop large peroxisomes that oxidize methanol and when grown on fatty acids, they develop large peroxisomes that break down fatty acids to acetyl-CoA and beta oxidation. Peroxisomes present in photosynthetic cells of higher plants in etiolated leaf tissue, coleoptiles, hypocotyls, tobacco stem and callus, ripening pear fruits, protozoa, brown algae, fungi, liverworts, mosses and ferns. Peroxisomes are also important in plants. Two different types have been studied extensively. First type is present in leaves, where it catalyzes the oxidation of a side product of the crucial reaction that fixes CO2 in carbohydrate. This process is called photorespiration because it uses oxygen and liberates carbon dioxide. The second type of peroxisome is present in germinating seeds, where it has an essential role in converting the fatty acids stored in seed lipids into the sugar needed for the growth of the young plant. The seedling uses this sugar until it is mature enough to produce them by photosynthesis. Because this conversion of fats to sugars is accomplished by a series of reactions known as the glyoxylate cycle. These peroxisomes are also called glyoxysomes. In the glyoxylate cycle, two molecules of acetyl-CoA produced by fatty acid break down into the peroxisome, are used to make succinic acid, which then leaves the peroxisome and is converted into glucose. The glyoxylate cycle does not occur in animal cells, and animals are therefore unable to convert the fatty acid in fats into carbohydrates. Import of protein in peroxisomes. A specific sequence of 3 amino acid located at the C terminus of many peroxisomal proteins functions as an import signal. Other peroxisomal proteins contain a signal sequence near the N terminus. If either of these sequences is experimentally attached to a cytosolic protein, the protein is imported into peroxisomes. At least 23 distinct proteins called peroxins participate as components in the process, which is driven by ATP hydrolysis. Oligomeric proteins do not have to unfold to be imported into peroxisomes, indicating that the mechanism is distinct from that used by mitochondria and chloroplast, and at least one soluble import receptor, the peroxin PEX5, accompanies its cargo all the way into peroxisomes, and after cargo release cycles back out into the cytosol. These aspects of peroxisomal protein import resemble protein transport into the nucleus. The importance of this import process and of peroxisomes is demonstrated by the inherited human disease Zellweger syndrome, in which a defect in importing proteins into the peroxisomes leads to a severe peroxisomal deficiency. These individuals whose cells contain empty peroxisomes have severe abnormalities in their brain, liver and kidneys, and they die soon after their birth. One form of this disease has been shown to be due to a mutation in the gene encoding a peroxisomal integral membrane protein, the peroxin PEX2, involved in protein import. A milder inherited peroxisomal disease is caused by a defective receptor for the N-terminal import signal. Most peroxisomal membrane proteins are made in the cytosol and then insert into the membrane of pre-existing peroxisomes. Thus, new peroxisomes are thought to rise from pre-existing ones by organelle growth and fission, as mentioned earlier for mitochondria and plastids, and as we describe for the endoplasmic reticulum. Let us have a look at functions of peroxisomes. Peroxisomes are involved in many types of biochemical activities. Let us discuss each of them one by one. Oxidative metabolism or hydrogen peroxide metabolism. Peroxisomes contains one or more enzymes like D-amino acid oxidase and urate oxidase that use molecular oxygen to remove hydrogen atoms from specific organic substrate, that is R, in an oxidative reaction that produces hydrogen peroxide. You can have a look at the reaction. RH2 plus O2 gives rise to R plus H2O2, that is hydrogen peroxide. Catalase forms 40% of total peroxisome protein. It catalyzes hydrogen peroxide generated by other enzymes in the organelle to oxidize a variety of other substances including alcohol, phenols, formic acid and formaldehyde by peroxidative reaction. We can have a look at the reaction where hydrogen peroxide reacts with R-H2 and leads to formation of R- plus 2 molecules of water. This types of oxidative reaction is important in liver, kidney cells whose peroxisomes detoxify various toxic molecules that enter bloodstream. Almost 25% of the total ethanol consumed in oxidized to acetaldehyde in this way. In this way, when excess hydrogen peroxide accumulates in cell catalase, converts hydrogen peroxide into water. We can have a look at the reaction where 2H2O2 leads to the formation of 2 molecules of water and oxygen. Most of cytosolic hydrogen peroxide produced by mitochondria and endoplasmic reticulum membrane, also produced by a cytoplasmic matrix by hydrogen peroxide producing enzymes. Catalase deals with high amount of hydrogen peroxide generated by the peroxisomes. Other enzymes like glutathione peroxidase capable of metabolizing organic hydroperoxides and also hydrogen peroxide in cytosol and mitochondria. Superoxide anion production in cytosol and mitochondria is regulated by SOD, that is superoxide dismutase. All these enzymes present in higher levels in aerobic tissues. A relationship is there between peroxides and free radicals like O2 with aging. These radicals act on DNA molecules and causes mutation. Free radical can also cause lipid peroxidation and protein denaturation. Hence, SOD and catalase including vitamin E keeps the cell healthy. Glycolate cycle. Peroxisomes of plant leaf contain catalase with glycolate pathway. Enzymes like glycolate oxidase, glutamate glyoxylate, serine glyoxylate, aspartate alpha ketoglutarate, aminotransferase, hydroxyl pyruvate reductase and malic dehydrogenase. Also contains NAD, NADP, FAD coenzymes. Glycolate cycle brings formation of amino acid, that is glycine and serine, from non-phosphorylated intermediates of phosphosynthetic carbon reduction cycle, that is glycerate to serine, glycolate to glycine as well as serine in sequence of reaction that involves cytosol, mitochondria, peroxisomes and cytosol. It serves as generator of nucleic acid biosynthesis precursors. Photorespiration. In green leaves, peroxisomes carry process called photosynthesis. It is light stimulating production of carbon dioxide that is differing from carbon dioxide generated by mitochondria in dark. Glycolic acid binds with oxygen and forms glycolate and hydrogen peroxide. This glycolate binds with hydrogen peroxide, leads to the formation of formate plus carbon dioxide and water molecules. In photorespiration, glycolic acid production of photosynthesis, released from chloroplast and oxidized into glycolate and hydrogen peroxide by peroxisome enzymes, glycolic acid oxidase. Later, glycolate is oxidized into formate and carbon dioxide. Glycolic acid is synthesized in chloroplast in presence of light. The next is beta oxidation. Peroxisomes of red liver cell contain beta oxidative enzyme for fatty acid metabolism. Fatty acyl-CoA is converted to acetyl-CoA. This conversion uses molecular O2 and NAD as electron acceptor. Acetyl-CoA formed migrates to mitochondria, where it enters into acetic acid cycle, that is TCA or Krebs cycle. If acetyl-CoA remains in cytosol, it is converted into fatty acids and then to neutral fats. In peroxisomes, dehydrogenase reacts directly with oxygen and generates hydrogen peroxide. Mitochondria have no catalase and cannot deal with hydrogen peroxide formed. Peroxisomes face no such problem. Let us have a look at role of plasmalogen in nerve cells. Plasmalogens are very abundant in the myelin sheaths that insulate the axons of nerve cells. They make up some 80 to 90% of the myelin membrane phospholipids. In addition to ethanolamine, head group and a long chain. Deficiency of plasmalogens causes profound abnormalities in the myelination of nerve cells, which is one reason why many peroxisomal disorders leads to neurological disease. Other functions. Peroxisomes contain D-amino acid oxidase present in mammalian liver and kidney, but is absent in other cells. This enzyme is present to oxidize D-amino acid that is found on cell wall of bacteria. Uric acid oxidase, that is also called urease enzyme, present in the peroxisome, is important in catabolic pathway that degrades purine. Peroxisomes can adapt to changing conditions. For example, yeast cells grown on sugar have small peroxisomes, but when yeast grown on methanol have large peroxisomes to oxidize methanol. When grown on fatty acid, have more large peroxisomes to break down fatty acids to acetyl-CoA. Let us have a look at biogenesis of peroxisomes. Initially, it was found that peroxisomes is formed by budding of the endoplasmic reticulum, while content of matrix is imported from the cytosol. But new peroxisomes arise from pre-existing one, being formed by growth and fission of old organelles parallel to mitochondria and chloroplast. Thus, peroxisomes are collection of organelles with constant membrane and variable enzymes. All structural and elastic protein is encoded by nuclear genes and is synthesized in cytosol, for example, catalase, that is tetrameric heme containing protein. Catalase and many other enzymes are having a single sequence of 3 amino acids, which is located near their carboxyl end and direct them to peroxisomes. Peroxisomes have signals on cytosolic surface to identify its protein. All membrane protein of peroxisomes is imported from cytosol. Lipid require to make new peroxisomal membrane also came from cytosol carried by phospholipid transfer protein from site of synthesis to endoplasmic reticulum membrane.
Now let us summarize the given topic. Peroxisomes are specialized for carrying out oxidative reactions using molecular oxygen. They generate hydrogen peroxide, which is utilized for oxidative purpose and destroy the excess H2O2 by means of the catalase enzyme. Peroxisomes also have an important role in the synthesis of specialized phospholipids required for nerve cell myelination. Like mitochondria and plastids, peroxisomes are thought to be self-replicating organelles. Because they contain no DNA or ribosomes, they import their proteins from the cytosol. A specific sequence of 3 amino acids near the C-terminus of many of these proteins functions as a peroxisomal import signal. The mechanism of protein import is distinct from that of the mitochondria and chloroplast and oligomeric proteins and can be transported into peroxisomes without unfolding. They are involved in other various functions like glycolate cycle regulation, beta oxidation, including oxidative metabolism. Thank you.